All right, so now we're going to start talking about the nervous system and nerves and nerve impulses and how all that kind of works in connection with, um, and I think you'll see there's some similarities from what you learned from the muscles that kind of builds up on that. This would be, I believe, chapter 7 if you're following the textbook. Please realize getting any type of textbook would be helpful. Even a general biology textbook that talks about how nerves work would be helpful. So let's go ahead and get started. So obviously, as you are, I'm sure, well aware of, the nervous system is made up of neurons. Neurons are specialized cells that can transmit information. They are excitable. They are able to communicate to one another and form a network. And so here is what we would call our classical motor neuron that you can see on the right. So this is, with my pointer, I'm pointing at it. The actual neuron is what we're seeing here, not these cells, these little white cells that are surrounding it. We'll get to those in a moment. But the actual neuron is, here's the cell body of the neuron, for instance. And it has dendrites, which are like little processes that extend off of the main part of the cell body. And then you see it kind of narrowed down. We call that an axon hillock. And then it continues traveling down to what we call an axon, ending in axon terminals, which is multiple ones you can see. If you recall from the muscle physiology lab, the axon terminals can synapse, remember the little space, they can synapse onto muscle cells at the motor end plates, or they can actually synapse onto other nerve cells where their dendrites are located usually, but other places as well. Inside the cell body, you'll see that there's a huge nucleus, and inside the nucleus, this little purple spot is called the nucleolus. The nucleolus is where ribosomes are made, and RNA is, ribosomes are made up of RNA and help make proteins. Like the muscle cells, nerve cells need to make a lot of proteins, and so they have a big nucleus. They also have a pretty substantial cytoskeleton that'll travel down the neuron. Now, these cells, while not necessarily really big in size, are actually really long, just like muscle fiber cells can be rather long. A nerve cell can travel from your back and innervate the muscles in your calf, so these can be really long. Now, surrounding the axon is a special group of cells we call Schwann cells. And each one of these little cells, and we'll get to it a little bit more later, make up what they call the myelin sheath. And we'll get into more of that, but those are important cells of the nervous system called glial cells. They are the support cells of the nervous system. So information travels from the dendrites through the cell body and down the axon to the axon terminals where neurotransmitters are released. And when I say information, I'm talking about a nerve impulse and ultimately depolarization. This will make a little bit more sense as we go along in the lecture, but if you recall from the muscle physiology, depolarization is when sodium enters into the cell. Well, that is also true for the nerve cells. Now this represents a motor neuron. However, sensory neurons also are part of your nervous system, but they would usually tend to be a lot shorter and found obviously in areas where you're doing sensing. of the environment, whether they be in your eyes or your skin, just about any place you can imagine. And their diversity and appearance is quite a bit different than these motor neurons, which are part of innervating muscles. And innervate, again, think about nerve, and then innervate is meaning that the nerve is, or the nerve cells coming in contact, at least up to the axon terminal, and then there's that little space where the synapse is located, or we call it the synaptic cleft. So in this case, nervous cells are incredibly important for animals. You don't find nervous cells in other types of organisms. You don't see them in plants, for example. And they're very important for behavior and for responding to the environment in a rapid manner. You could make an argument that some type of responsive behaviors may exist in plants, but it's not the same complexity that we see in animals, and nearly as quick. So here is examples of nervous systems of a variety of animals. Here we see a pretty simplistic nervous system in the sea anemone. If you don't know what this is, it's basically related to a jellyfish. And so they have a nerve network that travels throughout their body, but you don't really see a central nervous system. It's mostly made up of a nerve net. But we begin to see something that kind of looks like a central nervous system. Now, what is a central nervous system? A central nervous system, as you can see in this human, is the brain and spinal cord. The peripheral nervous system is everything outside of the central nervous system. So all these nerve fibers that you're seeing in the legs and in the arms and so forth are part of the peripheral nervous system. So this worm here has a central nervous system, simplified, of course, in comparison to our own. But you can see what they call these ganglion. And the ganglion are bunches of cell bodies and nerves that came together. They are actually found on the ventral side of worms and insects and so forth. We also see a rather complex nervous system with some complex invertebrate animals like the squid. They have a relatively large brain, easily among the invertebrates, that make them, in quotes, the intelligent invertebrates. They can do some very complex behaviors and communication with one another. They have large eyes as well. So anyway, that kind of gives you some ideas. Again, you won't see a central nervous system in a sea anemone. So again, the central nervous system makes up the brain and spinal cord, and the peripheral nervous system represents the nerves outside of the central nervous system. And even the support cells will be different. And again, a special term for support cells is glial cells. And we'll get into glial cells. These include things like myelin sheath that you probably have heard of. So to shorten the term central nervous system, we could write CNS, or the peripheral nervous system, PNS. So the central, or excuse me, this to kind of give you an idea of how the nervous system is classified. The peripheral nervous system includes both a sensory division and a motor division. So the sensory division, again, would represent your ability to pick up senses like sight, smell, touch, and hearing. And that information travels from either specialized organs like taste buds through the nervous system to the brain. We call that afferent, meaning it's traveling towards the central nervous system. Then the information gets processed And then the information travels back out through the motor output, out through the motor neurons. And we call that efferent, moving away from the central nervous system. The central nervous system, that is. So the peripheral nervous system, where information is traveling towards the brain and spinal cord, is the sensory neurons. The afferent division, and everything leaving the brain and spinal cord is the afferent or efferent. And again, the nerve cells will look quite a bit different. And these do two different examples. Now, the motor division can be broken up into two subdivisions: a somatic nervous system, which is voluntary, and an autonomic nervous system, which is involuntary. So again, this kind of goes along with what you remember from skeletal muscles and so forth, where you can move your arms, and while you might not have to necessarily think about it, it's still part of what we call voluntary. While the autonomic is usually outside of your conscious control. And this might include like regulating your heartbeat and so forth. So again, it's kind of giving you the idea of automatic. But I think you can have some gray in some of this. Now, if we look at the organization of the central nervous system and compare it to this model here, this box, you'll see the peripheral nervous system includes the sensory organs and then the sensory neurons sending information to the brain and spinal cord. And then that information gets processed and integrated and then sent back out a special group of motor neurons. And that's efferent. And of course, this can be broken up into the somatic, which is voluntary, or autonomic, which is involuntary. Again, this would be things like heart rate and breathing rate, and things that you just don't even give any time or thought into how to regulate or move. Like I could think about moving my hand consciously before I did so. That doesn't mean I can't have a reflex that causes, if you hit a knee with a little hammer, there's probably going to be some autonomic aspects to that. Now, the autonomic nervous system can be further broken down into the sympathetic nervous system and the parasympathetic. The sympathetic is our fright and flight responses. So if somebody clapped their hands really loud or something startled you, that would be part of the sympathetic nervous system. You'd be ready to run, you could feel jittery and excited. That is the sympathetic. The parasympathetic is the more relaxed, chilled mode. You're able to digest your food properly. Not to get too crass, but the parasympathetic nervous system works better for for sexual responses, while the sympathetic nervous system wouldn't, because you're dealing with more of the nervous and fright and flight responses. So like if you ate a meal and you're trying to digest it and you're freaking out because of something, it's going to be harder to digest that meal because all your hormones and stimulation is going to cause your heart rate to increase and so forth. Now, again, the nervous system is made up of specialized cells that we call neurons, nerve cells. And again, the nucleus is going to be full of metabolic activities. The cell body has a huge nucleus in it with a nucleolus. There's the fibers, as I mentioned before, that represent the dendrites where they receive information. We'll get into what it means to be a polarized cell, but the plasma membrane at rest is polarized, and that represents the relative charge. So the inside of a nerve cell, if you were to put an electrode into it and measure it, would actually have a negative electrical charge, while the outside of the cell, the extracellular fluid, would have a positive charge. And this is made up primarily of sodium and potassium and chlorides and a few things like that. So here is our neuron, and we got the cell and the cell neuron, and we got this long axon terminals, and then you can see a needle going into the nerve. And remember, this is a phospholipid bilayer. Hopefully you remember that from cell biology, since this is the cell membrane. And so let's pretend I'm a researcher putting a needle in and I want to measure the electrical charge. What I would find was that the inside of the neuron is more negatively charged. In fact, it could be anywhere from negative 60 millivolts to negative 80 millivolts, relative particularly to the outside. That is because the outside of the cell has a lot more sodium atoms. And if you're familiar with sodium atoms are positive ions from chemistry, meaning that they have an extra proton in comparison to electrons. Hopefully that's something that you're already familiar with. And so a lot of sodium atoms on the outside give the fluids on the outside a positive charge. Some of this, again, deals with evolution and the fact that cells evolved in saltwater environments where there was lots of sodium. So again, it's kind of evolution taking something like salt water, which is what's found that your cells are sitting in kind of, and utilizing it for a nerve impulse or muscle contraction. So again, there's a large nucleolus inside the nucleus, and it's important for making ribosomes. And the ribosomes, if you remember, attach to the endoplasmic reticulum, making a rough endoplasmic reticulum. Hopefully you remember that from cell biology. And so that means that the rough endoplasmic reticulum is important for making proteins because of all those ribosomes. When a messenger RNA comes up and hooks up to a ribosome, it's going to be translating proteins. Well, we call this Nissl substances, and that is where it's a specialized rough endoplasmic reticulum important for making proteins. Now, the neurofibrils are, again, made up of cytoskeletons that help maintain the shape of the cell. So that's, you can't see it here, but there's little fibers that make up the cytoskeleton. The dendrites, again, conduct impulses from other cells or from specialized sensory cells like taste buds, and then send that information, integrating it through the cell body down the axon hillock. And then the nerve impulse takes place and travels down the axon. Now there's different neurons. Again, this one is the multipolar neuron, also what you would call the motor neuron. And you can see the dendrites are associated with it. And then here's our axon, and then we have the axon terminals. And again, hopefully you remember what these little blue circles represent on this picture. That's correct. Those are glial cells, which are Schwann cells that make up the myelin sheath. And this is true for peripheral nerves. We'll get into this a little bit later. There's also bipolar neurons, which include this example here, where we have the cell body in the middle, and we have dendrites and axons, and it's very short. And so this is something you'd probably see in your eye for photoreception, along with the photoreceptor cells like rods and cones. Then we also have bipolar neurons. This was the bipolar neuron. This is the multipolar neuron. And then we also have unipolar. And in this case, the cell body extends on the outside. So see the cell body's in the center. And I don't see any myelin sheath on this picture. But over here, the unipolar, we do see myelin sheath, and we see the cell body is extended outwards. So it's kind of hard to tell where the dendrite begins and the axon forms. But again, this is also a peripheral nervous system as part of the sensory nervous system. So both of these cells, the bipolar neuron and the unipolar neuron, are part of the peripheral nervous system, but particularly in regards to the sensory nervous system that picks up taste, touch, stretch receptors even in your stomach. When you know you're eating too much, that's all part of that kind of nervous system. And so if you look at the sensory system here, you got your, again, your bipolar and your unipolar or pseudo-unipolar neurons. Then you have over here on the right side our multipolar neuron, which is part of which is a motor neuron that's sending information to muscles. So they're the effector neurons, and these are the receptive neurons on the left. Now, there's also this group in the middle called interneurons. And as the name might imply, they're in between these two sets of neurons. And they're found in the central nervous system, where the integration takes place for your brain to interpret, or your spinal cord to interpret information from the afferent neurons. And then your body's response will be coming down the efferent neurons, the effector neurons. And so here is a simple reflex arc setup. Here you have our peripheral neurons. Maybe it's picking up some information from your skin, or maybe there's a reflex in your knee that's getting hit with a hammer. That information will travel down the sensory neuron. And in this case, it looks like a pseudo-unipolar neuron or unipolar neuron. That information reaches the spinal cord, sends information down the associated neuron. I don't necessarily mean to quote, but remember it's going to be neurotransmitters being released at the synapse here. And then this neuron is going to make a decision that ultimately probably affects whether or not this motor neuron will send a signal back to the muscles to move or whatever. Now, most of the central nervous system, so the cell bodies, this is what the cell body looks like, right? Most of it is found in the central nervous system, while the fibers extend out of the central nervous system and join the peripheral nervous system. So when we talk about gray matter, we're actually talking about the cell bodies. And it looks gray because the neuron looks gray, while and it's unmyelinated. The myelinated part looks white when you look at a nerve fiber. So it's actually, the gray matter is made up of the cell bodies. So the nuclei is a cluster of cells that are found within the white matter of the central nervous system. So nuclei are a cluster of cell bodies within the white matter of the central nervous system. So if you were to slice open some spinal cord or some brain and you see a cluster of cell bodies, we'd call that a nuclei. However, if it's outside of the central nervous system, like in the peripheral nervous system, we'd call it a ganglia. So if we see some gray body that's outside of our brain and spinal cord, it's ganglia. But if it's inside, it's nuclei. And so basically neurons work by transmitting information to one another. So here's a dendrites of one nerve. The information will travel down the axon hillock, down the axon, reach the dendrites of the next neuron through these axon terminals. This little space is called the synapse, the synapse is the area, and the synaptic cleft is the actual gap where the neural transmitters will flow across. So again, information will travel from the one neuron to the next neuron. This one you should notice is they actually referring to it as presynaptic neuron. That means it's in front of the synapse. So this is the synapse, the space between this axon terminal and this neuron. So this is the presynaptic neuron in relationship to this neuron, which is the postsynaptic. And as you can imagine, you could have a neuron that is presynaptic, and then this one is postsynaptic, but this one postsynaptic neuron could be presynaptic to another neuron. So again, it's just showing you how it flows from one neuron to the next. And again, the synapse is the space between, that includes the axon terminals and the dendrites, and then the actual space is the synaptic cleft. So the information is traveling from this neuron down to these neurons. We're going to get into how a nerve impulse works, but that's not the only way that information travels down a neuron in the sense, what I call information. And so we're going to talk about an important aspect of how neurotransmitters are made and then travel down to the axon terminal. And so let's go ahead and talk about that. So here we have our neuron. We have a cell body. We have a nucleus. That dark blue here is probably the nucleolus. And outside of it is our nissl substances that are made up of the rough endoplasmic reticulum. As I understand, it's called nissl substances. And you'll see that those little dots represent ribosomes. And again, remember the central dogma of biology. The central dogma of biology, if you'd forgotten from a class, is that DNA that's made in the nucleus, or is found in the nucleus, it's the blueprints of cells. The DNA gets transcribed, and hopefully you remember this from a general biology class or cell biology class. The DNA gets transcribed into messenger RNA. The messenger RNA then leaves the nucleus, just floats out, travels to a ribosome, hooks up to a ribosome, and then gets translated, where amino acids come in and makes a protein. That's what's taking place at the rough endoplasmic reticulum, as you can see with my pointer now, with my cursor, or with my mouse pointer thing, whatever you want to call it. So you can see those little dots represent the rough endoplasmic, well, it's the rough endoplasmic reticulum, but those dots represent the ribosomes. And where were the ribosomes made? In the nucleus, the nucleolus it's found inside the nucleus. And so then when the proteins are made, and they call it peptides, because remember, peptides are made up of amino acids. So you have a peptide chain made up of a chain of amino acids. The peptides are formed, and they'll travel to the Golgi body. Now, what does a Golgi body do in a cell? Hopefully you remember this. It modifies and finishes up the packaging of the protein. Often these proteins are going to be neurotransmitters, like acetylcholine. And so how do we get those neurotransmitters or substances down this long axon? Because remember, an axon is really long. It can travel from your spinal cord down to your ankle, for instance, or further. So how do we get these substances to go down this axon? Well, what happens is the vesicle hooks up with some tubes that are made up of the cytoskeleton, and that's what these are. So these are protein tubes, kind of like actin. So think about actin for a moment. It might not be exactly actin, but it's a protein filament. And on the outside of these vesicles are little proteins that are kind of like myelin heads. Remember how myelin heads could grab and pull? Well, and I'll show you some video and picture to help you make sense of this. But these vessels have these little sticky hands and fingers that are around it that allow it to attach to this microtubule and actually walk the vesicle down the tubule. It's almost like an escalator or a sidewalk with these vesicles walking down the tubes. And how would they do it? With ATP, of course, fueling the protein conformational change. It's kind of like a hand or feet walking down this tube. When the vesicle reaches the axon terminal, which is the end of the axon, the vesicle can then be released and the neurotransmitters can go into the synaptic cleft. And then remember what the neurotransmitters can do? They can trigger the muscle to contract. So this is how the neurotransmitters are made and travel down the axon. We call that fast axonal transport. Now, the cell can also, once it's done with that neurotransmitter, recycle the neurotransmitters and use it for something else, or maybe waste products or whatever. They'll enter into a vesicle, and then the vesicle can walk itself back up to the cell body to be digested with lysosomes. Remember, lysosomes are a bag of digestive enzymes that can break down substances. Now, if you're not familiar with this, I'm hoping you remember this from cell body when you look at a picture like this. Notice when the vesicle comes along, and remember the vesicle is going to be like a cell membrane, it will attach to the cell membrane of the neuron. And then the vesicle will release its substances. We call that exocytosis. And if you're not familiar with that, you should look it up. That's something that every biologist should already know without me having to explain it at this point, if you've taken some biology classes. If not, look it up. But anyway, the cell membrane becomes part of the outside and the neurotransmitter is released. Now, if you remember endocytosis, the substances on this picture are coming in, making the vesicle. And then again, the vesicle will walk up the microtubule. Since it's going in reverse, we call that retrograde fast axonal transport. And then that vesicle will walk up, and again, the vesicle is like a bubble made of a cell membrane. It'll hook up with a lysosome, which is a cell membrane. And it's almost like two soap bubbles coming together and joining into one big bubble. And there, the lysosome enzymes can break down the neurotransmitters or whatever needs to be broken down. Here is a picture showing you how how a vesicle can walk down a microtubule or an organelle or whatnot can walk down these microtubules. They have these little feet-like projections that are kind of like the globular heads of myelin. So it's kind of like the muscles and their contractions, but in this case, it helps walk a vesicle down or organelle down to various parts of the cell. And what happens though, and I'm going to show you a video of that. I'm going to try to see if I can find something for you in a moment. But what happens when a nerve is damaged? It's been severed. Unfortunately, you can get severed with a cut with the blade, or you can be in an accident. What happens is, if the nerves can't heal itself, it's been stretched, it's one thing, but if it's actually ruptured, this portion of the axon will die, even if the cell body lives. The problem is that the muscles and stuff like this are no longer innervated. So information is no longer traveling, and so that part of your body becomes non-sensory wise, or the muscles don't work. There is some interest in trying to figure out how to solve that, maybe through stem cells, making new nerve cells. And they're also looking at the nose cones that help in figuring out where the axon terminal will travel. So on the front of an axon terminal during development, like as an embryo, as the nerve's stretching out, there'll actually be a cone of cells over the axon terminal, and it'll help attract the axon terminal to the muscle cells and attach. We call that innervation, innerving. we'll go back and look at fast axonal transport and so forth. Let's get into the support cells of the neurons. And then after that, we'll look at a video or two, and that'll be finishing it. So this part's still really important. The nervous system isn't just made up of neurons. It is also made up of these glial cells. And again, these are the support cells of neurons, and they're extremely important. If you didn't have myelin sheath, for instance, the nerve impulse would be dramatically slow and would not work. So you really want these glial cells. And different ones have different functions, and so we're going to get into that now. And so be sure to read this here. So we're going to hit on some major ones. Some are found in the peripheral nervous system. Remember, the peripheral nervous system is the parts of the nerves that are outside of the central nervous system, outside of the brain and spinal cord. And so these glial cells include satellite cells and Schwann cells. Satellite cells are cells that surround the cell body of a nerve. In this case, this looks like a pseudo-unipolar neuron. And so if you look at the pseudo-unipolar neuron, you can see the cell body here. You can see a nucleus, and then that little dot there is probably the nucleolus that makes the ribosomes and so forth. Well, these are cells on the outside of the cell body. And so you can see they each have their own little nucleus. That's what the little dot represents. And so they're surrounding the cell body. Then we also have another group of cells called Schwann cells. And so here you can see the Schwann cells are wrapping around the axon. And so these would be our myelin sheath. And between each of these myelin sheath is a little space that we call the node of Renevier. If I think I pronounced that right, but we'll check. What is the function of myelin sheath? We'll get into how it does it a little bit more, but it is to speed up a nerve impulse. And if you were to have your myelin sheath damaged or removed, which sometimes happen in diseases like multiple sclerosis, where your myelin sheath can be damaged, it can dramatically harm your nerve impulses from working rapidly. It's an autoimmune disease of some sort that's where your immune cells are destroying the myelin sheath. And it can cause, problems, paralysis, and so forth. I didn't really talk much about the satellite cells, but the satellite cells are providing protection to the neuron cell bodies and also probably involved in nutrient control as well. Nutrition and other factors are probably, I would guess, would be part of that as well. So again, these parts that make up these Schwann cells, they're almost like a string of beads on a string. The string would be the axon, and then the beads are the glial cells making up the myelin sheath. Now that's the peripheral nervous system. And here you can see a picture of some Schwann cells wrapping around the axon. So you can see the axon being wrapped around, so slowly being surrounded. And this will happen, again, it almost looks like beads going down a string. Have you ever made a bead necklace? And when you look at the myelin sheath of the Schwann cells, it almost looks like a flat pancake wrapping around a sausage link. Have you ever had pigs in a blanket? It's kind of like that. Here's another picture showing you Schwann cells. And so you can see the axon is here, or excuse me, the nucleus is here, the axon is the blue, so that's what's shown in this picture here. I called it the string, or it'd be the sausage. We were talking about my other example. the pigs in the blanket. And then the axon, the Schwann cells are wrapping around the axon multiple times. See that arrow? It's showing you're being wrapped around multiple times. And again, this is cell membrane. And what are cell membranes made up of? Hopefully you remember phospholipids. Good. So you can see it being surrounded. And then the little gaps between each one of these Schwann cells is that node or renovere I was telling you about. And we'll explain more about how that is important later. Over here, you can actually see an electron micrograph of a Schwann cell or axon being surrounded by myelin sheath. So here you can see myelin sheath surrounding it in multiple layers. And then here you can see your axon. So that's the peripheral nervous system. Now, what about the central nervous system? Well, it has its own set of glial cells. So remember, the glial cells are support cells. In the peripheral nervous system, we had Schwann cells that make myelin sheath. And we had, let's go back and look. We had satellite cells that surrounded the neuron cell body. Now, myelin sheath, as I mentioned before, is made of Schwann cells, but it's not just Schwann cells. Myelin sheath can also be made up of another group of glial cells called oligodendrocytes. But this is the big difference. Oligodendrocytes are found in the central nervous system. So the glial cells of the central nervous system is what we're talking about now. And so here we have our oligodendrocyte, and there's another big difference. The oligodendrocyte surrounds multiple axons, because remember the Schwann cell wrapped around one axon, like beads. Well, in this case, this oligodendrocyte is surrounding multiple axons and surrounding them. As you can see in this picture, here's our nerve fibers of this. So there's multiple axons that this oligodendrocyte is surrounding and bracing in a sense too, helping hold them in place. So the oligodendrocyte is able to surround multiple axons. Now, the astrocytes are another group of glial cells that we find particularly in the brain. And they have an important role in forming the blood-brain barrier. So the astrocytes will actually surround capillaries, blood capillaries. Remember, capillaries are little thin tubes for where blood can transport oxygen and so forth. Well, the astrocytes form the blood-brain barrier to help keep toxins out of the brain and also brace and surround parts of the axon near the cell body. And so that's what makes up the blood-brain barrier. Obviously, the blood-brain barrier doesn't keep all toxins out. We know, for instance, alcohol is blocked. Or excuse me, alcohol is not blocked because alcohol is lipid philic. So remember, a cell membrane is made up of lipids, and alcohol can cross that lipid barrier easily. So again, so we talked about so jumping back, peripheral nervous system, we have Schwann cells, and we have satellite cells. The central nervous system, we have oligodendrocytes and astrocytes, and we have a couple more. Just remember, oligodendrocytes and Schwann cells both make myelin sheath. So the myelin sheath in the peripheral nervous system is made up of Schwann cells, and the myelin sheath is made up of oligodendrocytes in the central nervous system. We also have microglia, which are a group of spiders that are part of the immune system. And they run around, they're phagocytes. That means that they're cells that are going to walk around and eat bacteria and other things that aren't supposed to be in the nervous system. So we call those phagocytes. And they'll dispose of debris, and they'll eat, and they'll be part of the immune system and so forth. We also have epithelial cells that are made up of cells that provide the fluids needed to make the cerebral spinal fluid. And so the cerebral spinal fluid will travel around and provide a cushion to the brain. So if you rattle your brain, the cerebral spinal fluid is helping cushion your brain from hitting your skull and so forth. I'm a cancer survivor, so I actually survived and thankfully survived non-Hodgkin's lymphoma. I actually had Burkitt's lymphoma, non-Hodgkin's lymphoma. It's called atypical Burkitt's lymphoma. And anyway, to make a long story short, when I was receiving chemo, it would actually be injected into my spinal cord, so I'd have a spinal tap, and some of the nasty chemo would be put to my brain and spinal cord directly. And make a long story short, when they put the needle in, it would go into the cerebral spinal fluid. But sometimes that cerebral spinal fluid would leak. And I would lose quite a bit of that fluid that was cushioning my brain. And I can tell you from having a drip that it's a terrible headache to not have that cerebral spinal fluid. The cerebral spinal fluid is really important for protecting your brain and cushioning it. Some of those epithelial cells that help make up the cerebral spinal fluid, I believe, can also have some stem cell function. I don't know if they can go as far as making new neurons, but they do have some potential. Sometimes when you can get spinal taps for other reasons too, those painkillers for during delivery. So anyway, if the cerebral spinal fluid leaks, you're in for a big, nasty headache. So this picture here is the overall view of glial cells. Again, the peripheral nervous system is made up of satellite cells that support cell bodies and Schwann cells that help form the myelin sheath, and they provide neurotrophic factors, which means they're just substances that are probably influencing nerve responses and growth or whatever. It could be a lot of things. And then of the central nervous system, we have epithelial cells that help form the cerebral spinal fluids and may have some stem cell function. We have microglia cells, which are part of the immune system that are scavengers, or maybe they have some role in getting rid of nasty things that aren't supposed to be there, obviously. And then astrocytes make the blood-brain barrier. They help brace the brain and nerve cells in regards to capillaries, so they have a bracing factor. They can also secrete neurotropic factors that influence the cell's responses. They also regulate potassium and neurotransmitters. And then last but not least, the oligodendrocytes make up the myelin sheath of the central nervous system. And so this picture here kind of depicts them all involved with each other. Here we got our inner neurons and which are blue, oligodendrocytes and epithelial cells and so forth. Our next lecture, we'll get into nerve impulses. Let me see if I can show you fast axonal transport and a few different things from whatever is available on YouTube for a moment. Okay, so we've been talking about, if you go a little bit further back, we talked about how things move down these microtubules. And so we call that fast axonal transport. It might also be called anterior axonal transport and then retrograde axonal transport. So going down the axon to the axon terminals, and then retrograde mean going back the opposite direction. I mentioned how it can take vesicles down there. You can also take organelles down the tubules. So those are different things that can be transported up and down these microtubules. And again, it's going to be proteins that are going to be attached to the vesicles or tubes or whatever that are being transported. And again, it could be organelles, vesicles, tubes. I should also mention, and this is something I want you to write in your notes if you haven't already, what's the other way that substances move in neurons or cells in general? Well, diffusion. So things can kind of, or microfluidic fluids can kind of push things around. So if you think about a nerve cell being kind of filled with fluids, right? Little substances can kind of float around and move around from usually from areas of higher concentration to areas of lower concentration in the neuron. But when we're talking about big substances and doing it quickly, it's again this axonal transport. And this animation will kind of give you an idea. So here's our microtubes. And then we have this connective protein substance thing called tauus. I'm not sure if that's exactly how I pronounce it, but you can see it's kind of like these microtubules are made up of probably some type of protein. I don't know if it's actin, but it's kind of has that shape, like what we've seen on actin. And so then if you look in, so here's the tau protein. I'm not sure if I said that quite right. But here's, watch this right here. This could be a vesicle. This could have been an organelle. And then here you can see it's kind of like a globular head that we saw in muscles. And I presume ATP is being utilized to help move and function as motor, to move along. It's kind of like a motor protein. It's like a motor, right? So, but it's, or feed is what I keep calling it. But here you can see it walking down the tubule. So these substances are moving from the cell body down the axon to the axon terminal, and then they, of course, can do retrograde and go back the opposite direction. This is showing you what happens if you get nerve damage, like I was telling you earlier. So now look what happens when you get a nerve damage. The microtubules can no longer be utilized to bring the substances down. So what happens to the axon terminal? If it isn't repaired, it's going to die. Not necessarily the whole neuron, but that first part of it. But let's take a look at that animation one more time. Here's the substances walking up and down them. And again, it looks like globular heads to me that was found along with myosin. So these kind of look like myosin heads to me. It might be a different protein, but I wouldn't be surprised if it's a similar kind of idea. So that concludes the first part of nerves and our nerve talk. We got into glial cells. We got a little bit into fast axonal transport. So for your review, I would like it for you to include talking about fast axonal transport. I would like it if you mentioned something about glial cells, and I would also think it's pertinent to talk a little bit about the anatomy of a cell. And so, again, summarize that as single space typing. It should probably be every bit of half a page or more. If you go a lot less than half a page, you do lose some points. And I would like it if you well, I would like it if you explained fast axonal transport. So there's a diagram that I showed you. It's going to be on slide. It should be apparent, but it should be on slide 18. So slide 18 will show you. Slide 18 will show you the neuron, and it'll explain fast and slow axonal transport. I would like you to draw that and diagram it and explain it along with your summary. And that will give you the points for participation in this lecture. After we start the next lecture, we'll get into how a nerve impulse actually works. But anyway, that gives you an introduction to how the nervous system works. And don't forget the unsung heroes of the nervous system are those glial cells. They really rapidly increase a nerve impulse, provide a home of some sort for the nervous system. I would say, I'm not sure what the percentage breakdown is, but a big part of your brains is these glial cells. And of course, that's also true for a lot of your peripheral nervous system. Anyway, I hope you got something from this lecture and look forward to seeing you in the next lecture. Don't forget to start focusing on learning all you need to for the muscle section, as you will get an oral exam most likely, or take home depending on, or take home an oral, or it'll be in class if we don't have COVID problems. But obviously, for this, at least this first run, it's during our COVID time. Anyway, thank you so much for paying attention.